In this video, we will be discussing how the brain is protected. You can find information on this topic in Chapter 12 of your textbook. Your learning objectives are as follows. Describe the following and explain how each protects the brain. The skull, the meninges, cerebral spinal fluid, and the blood-brain barrier. Describe the circulation of cerebral spinal fluid. And finally, describe hydrocephalus, its cause, and its treatment. This image from your book, or similar to an image in your book, shows a frontal section of the brain. And here you can see the first line of defense from physical damage to the brain is, of course, the skull. The skull would absorb some of the energy from a physical blow to the head. So if someone were to hit you with on the head with an object, or if you were to fall on your head, some of that energy would be absorbed by the skull and hopefully not impact the brain. Deep to the skull, you have the meninges. If you recall, the meninges include 3 layers of connective tissue membranes. The outermost is called the dura mother, which literally means hard mother. It's actually composed of 2 separate layers, although you don't have to know the names of those layers. This is the thickest of the 3 meninges. It provides a physical barrier against infection. Notice that the inner or deeper layer of the dura mater actually dives into the cerebral fissure, the longitudinal fissure. Deep to the dura mater is the arachnoid. Arachnoid mater is so named because it sort of looks like a spider's web. Notice that there is the space below the arachnoid, and this is called the subarachnoid space. The subarachnoid space is filled with cerebral spinal fluid, which we'll talk about in just a few moments. And also notice that there are also a large concentration of blood vessels. The third and deepest of the 3 meninges is the pia mater, and the pia mater is very thin. Pia mater literally means gentle mother, and it clings tightly to the cerebral cortex. So these 3 meninges, along with the subarachnoid space, provides a further cushion and physical barrier to infection of the brain. Let's talk now about that cerebrospinal fluid. You are looking at an image from your textbook showing an anterior view of the brain and a lateral view of the brain. There are 4 chambers, or I guess you could call them cavities, within the brain, and those cavities are called ventricles. You've got 2 lateral ventricles. Sometimes they're referred to as lateral horns because they sort of resemble a horn. And they are connected to the 3rd ventricle via small passageways called interventricular foramen. And then the 3rd ventricle is connected to the 4th ventricle by the cerebral aqueduct. Just to give you a little orientation, remember that here you would have the thalamus. Remember, the thalamus surrounds the 3rd ventricle. And down here in the 4th ventricle, this would be, anterior to the cerebellum. Let's now look at a sagittal section of the brain and spinal cord. In addition to cerebral spinal fluid being found in the ventricles of the brain, it is also found in the subarachnoid space. So cerebral spinal fluid is made at the choroid plexus, and there's one in each ventricle. Here's the ones of the third ventricle and fourth ventricle. There are passageways, which we'll talk about in a moment, from the ventricles into the subarachnoid space. Remember, the subarachnoid space is the space below the arachnoid, which completely surrounds the brain, and it also surrounds the spinal cord. The significance of this is that the brain literally floats in cerebral spinal fluid. The significance of this is that the buoyant properties of the cerebral spinal fluid prevent the brain from being crushed under its own weight, because it literally reduces the weight of the brain by 97%. If not for the cerebral spinal fluid, the underside of the brain here would be crushed on under the weight of the rest of the brain. The cerebral spinal fluid found here between the brain and the skull also provides additional protection from blunt force trauma. So being hit in the head or falling on your head, some of that energy is going to be absorbed by the cerebral spinal fluid. Let's look now at how the cerebral spinal fluid is formed and the composition of the cerebral spinal fluid. So, where does cerebral spinal fluid come from? Cerebral spinal fluid is produced by a knot of capillaries called the choroid plexus. In the lateral horns, the third ventricle, as well as the fourth ventricle, you have a knot of capillaries, and these capillaries are covered by the ependymal cells. Remember, the ependymal cells are those glial cells that have cilia. Those cilia are used to circulate the cerebral spinal fluid throughout the chambers or ventricles. Cerebral spinal fluid is made from the plasma. So here we have blood flowing through the capillaries, and specific substances are allowed to cross the wall of the capillary, move through the ependymal cells, and into the ventricles. This process excludes most substances, only allowing specific substances to cross. At the same time, waste products that are in the cerebral spinal fluid move in the opposite direction across the ependymal cells, across the wall of the capillary, back into these capillaries of the choroid plexus. Since the cerebral spinal fluid is produced from plasma, its nutrient composition is similar to that of plasma, although typically the ion concentrations are lower than that of plasma. So the cerebral spinal fluid contains glucose, oxygen, some lactic acid, a little urea, which is a waste product produced by cells of the body, cations such as sodium, potassium, calcium, and magnesium. So the cerebral spinal fluid does provide some nutrients to nervous tissue. Let's take a moment to describe the circulation of cerebral spinal fluid, starting with its production in the choroid plexus of the lateral ventricle and ending with its reabsorption back into the venous blood supply, or in other words, the veins of the cardiovascular system. The choroid plexuses of the lateral ventricles produce the cerebral spinal fluid. It circulates from the lateral ventricles into the third ventricle through the interventricular foramina. The cerebral spinal fluid, now in the third ventricle, is joined by additional cerebral spinal fluid produced by the choroid plexus there. Cerebral spinal fluid is then circulated through the cerebral aqueduct into the fourth ventricle. In the fourth ventricle, there is an additional choroid plexus that adds additional cerebral spinal fluid. It is at that point that the cerebral spinal fluid leaves the ventricles, or the third ventricle, and enters the subarachnoid space through the lateral apertures and one median aperture. Cerebral spinal fluid then circulates through the subarachnoid space and eventually leaves the subarachnoid space through the arachnoid villus, which allows it to be reabsorbed into the venous blood supply. The production of cerebral spinal fluid usually equals the reabsorption of cerebral spinal fluid back into the blood supply. So cerebral spinal fluid is continuously being made and it's continuously being reabsorbed back into the blood circulation at equal rates. Let's examine how cerebral spinal fluid circulates through and around the brain. We're going to start here in the lateral ventricles where the choroid plexus produces cerebral spinal fluid. That cerebral spinal fluid then passes through the interventricular foramina into the third ventricle. In the third ventricle, another choroid plexus adds additional cerebral spinal fluid. Cerebral spinal fluid then passes through the cerebral aqueduct into the fourth ventricle. There we have another choroid plexus that adds additional cerebral spinal fluid. From the choroid plexus, rather from the fourth ventricle, cerebral spinal fluid passes through 3 apertures, 2 lateral and 1 medial, into the subarachnoid space. Cerebral spinal fluid then circulates through the subarachnoid space around the brain as well as around the spinal cord and is returned to the blood supply at the arachnoid villi. Normally, production of cerebral spinal fluid equals the reabsorption of cerebral spinal fluid back into the veins of the cardiovascular system. Sometimes there is a reduction in the reabsorption of cerebral spinal fluid back into the blood supply. This can be caused by a blockage of the apertures that allow cerebral spinal fluid to move from the fourth ventricle into the subarachnoid space. A reduction in the flow of cerebral spinal fluid into the subarachnoid space leads to a reduction in the reabsorption of cerebral spinal fluid back into the blood. The result is a buildup of cerebral spinal fluid. With an increase in the volume of cerebral spinal fluid, that increases pressure that the cerebral spinal fluid puts on the nervous tissue, and that can damage and destroy nervous tissue. Hydrocephalus is a medical condition that requires immediate attention. In an infant or toddler where there are still spaces between the cranial bones, it allows the cranium to expand and provide some relief of the pressure. Typically, one treatment of hydrocephalus is to insert a shunt that will drain the cerebral spinal fluid into a vein of the neck. Another treatment is to make a hole directly between the third ventricle and the subarachnoid space. This allows cerebral spinal fluid to therefore move into the subarachnoid space and eventually be reabsorbed via the arachnoid villus into the blood. So how does the cerebral spinal fluid protect the brain? Well, we already talked about how its buoyancy prevents the brain from being crushed under its own weight. We also talked about how the cerebral spinal fluid acts as a shock absorber as it separates the brain from the cranial bones that surround it. We talked about the composition of cerebral spinal fluid, and what I didn't mention is the concentration of electrolytes provides a very good environment to support the production of action potentials and the normal neuronal signaling that occurs between neurons. And lastly, the cerebral spinal fluid allows for an exchange of nutrients and waste products between the blood and the nervous system. The brain is well supplied with blood. Even though it only represents about 2% of adult body weight, the brain receives about 15% of the body's total blood supply or flow. It consumes a staggering 20% of the body's total consumption of oxygen and glucose. So blood supply is critical to maintaining the proper functioning of the brain. At the same time, it can also be a source of danger because anything that makes it into the blood could potentially make it to your brain. So toxins from contaminated food, fluctuations in electrolyte levels due to eating food that is high in a particular electrolyte, bacteria and viruses that make it into the blood, again, due to injury or perhaps contaminated food, and white blood cells, which, while fighting and destroying microorganisms that make it into the body, can also cause damage to tissues. That potentially is a source of danger that blood supply presents. This is where the blood-brain barrier comes into play. The blood-brain barrier is composed of the least permeable capillaries of the body. The capillaries, the walls of the capillaries are composed of cells that have tight junctions between one another. If you recall, tight junctions are impermeable junctions. They don't allow substances to pass between cells. In addition, astrocytes, those glial cells found in the central nervous system, they have perivascular feet that wrap around the outer surface of the walls of the capillaries and reinforce those tight junctions. This image from your book illustrates the structure of most capillaries seen here on the left versus capillaries of the blood-brain barrier. So as you can see, the wall of a capillary is composed of simple squamous epithelial cells, and notice that there are gaps or spaces between adjacent cells making up the wall of the capillary. So substances can cross the cells themselves, but they can also pass between cells in most capillaries of the body. Let's contrast capillaries of the blood-brain barrier. So first of all, there are tight junctions between adjacent cells, so nothing can cross between. Substances can only enter and exit the capillaries of the brain by entering on one side of these cells and exiting on the others. This excludes most molecules except those that are lipid soluble from entering the brain. Recall that lipid soluble substances can pass across the plasma membrane of cells. So lipid substances can easily cross the blood-brain barrier, not so much for water-soluble substances. As a result, the blood-brain barrier is permeable to few charged substances, namely water and glucose. Other charged substances, such as creatinine, ions, and urea, they can cross, but they cross relatively slowly. Now, ions would be crossing mainly from the blood into the interstitial fluid. Creatinine and urea, which are waste products, would be going the opposite way. The blood-brain barrier is not very permeable to antibiotics at all. Most antibiotics are water-soluble. In stark contrast, lipid-soluble substances and other non-polar substances can cross the plasma membrane relatively easy. So alcohol, which is a lipid-soluble substances, most anesthetics, and oxygen and carbon dioxide can cross the blood-brain barrier with ease. The blood-brain barrier is therefore very effective at keeping harmful substances that make their way into the blood from the brain, as long as they're water-soluble, because water-soluble substances can't cross the wall, the plasma membranes of the cells which make up the wall of the capillaries. Lipid-soluble substances that are harmful that get into the blood can cross still. But the blood-brain barrier is also a double-edged sword, because if you have an infection in the central nervous system that you're trying to treat, it's also difficult to deliver medication into the central nervous system via the blood, because most antibiotics are also water-soluble and therefore do not readily cross the blood-brain barrier. It is important to note that certain diseases and certainly physical damage to the central nervous system can compromise the blood-brain barrier, allowing harmful substances that make their way into the blood to get into the interstitial fluid of the central nervous system.